The purpose of this experiment is to determine if earthworms in soil contribute to plant germination and growth. According to Groningen, earthworm activity affects biotic and abiotic soil properties, which has an effect on plant growth. Using meta-analysis, it has been shown that on average, earthworm presence in agroecosystems leads to a 25% increase in crop yield and a 23% increase in above-ground biomass. Groningen's experiment also determined that positive effects of earthworms become larger when more residue is returned to the soil, but disappear when nitrogen availability levels are high.
We predicted that the trays containing 2 worms per plant would germinate at a higher rate than those with no worms and 1 per plant. We also predicted that the trays containing 2 worms per plant would grow more than those without or with only 1 per plant.
In order to complete the experiment, we needed 60 earthworms, 60 radish seeds, topsoil, plant trays, a scale, and a caliper.
To set up our experiment, we put topsoil into the trays, and then we measured the masses of 60 individual worms and got their average. Then we placed 10 seeds in each tray, and we labeled the location from each seed. Then after that, we placed 10 worms in 2 trays by burying them. Then in the next 2 trays, we placed 20 worms in them. Then for the next remaining trays, we left them without worms, and then we put them in the greenhouse to grow.
The plants were watered 3 to 5 times a week, depending on soil dryness. Any dead worms will be removed and replaced with new worms.
After the height data was recorded, we uprooted the plants and we placed them in the oven for 24 hours to dry out. We removed the dry plants and we averaged their dry weights to determine which plants grew the most.
On average, the seeds in the trays with no worms had the lowest germination rate of 50%, while the seeds planted in the trays with 2 worms per seed had the highest germination rate of 75%.
At the end of week 2, more than half of the plants had fallen over and begun necrosis in the trays with none and 1 worms per plant, and about 8 plants in the trays with 2 worms per plant had fallen over and began necrosis. By week 3, only 3 plants remained alive in the trays with no worms, 3 plants remained alive in the trays with 1 worm per plant, and 12 plants remained alive in the trays with 2 worms per plant.
In conclusion, the trays with the highest number of worms, of 2 worms per plant, had the highest average growth. The tray with the highest number of worms also had the highest dry weight average. The conclusions derived from our experiment showed support for both of our initial hypotheses. The data supported the idea that worms do contribute to plant growth and germination rates in a positive manner. The more worms present, the higher the germination rate and the more the plants grew.